So we are going to learn about protein synthesis today. And notice that I didn't say okay when we first started. Good.
I said okay 3 times before that I counted.
Had to get them out. So, we're going to learn about this process, going from DNA to a protein. What are proteins made of?
Amino acids.
Amino acids. Where are proteins? Ribosomes. Very good. So we can infer that this is a, what is this right here? A ribosome. A very poorly drawn ribosome. Ribosome. So, let's go through this. I'm looking over the notes over here. You don't have to, just follow along, make sure you have everything written down correctly. You said that on my printer. I moved your desk, I moved your stuff right here, so you weren't in frame. Sound good? So what we're talking about with protein synthesis here, and am I all in frame? So, you still in frame or no?
Yes.
Okay. Nice. The process of copying DNA and using that copied information to make protein. This is all notes you should already have. I'm just reviewing this. I'm going to go through this quickly. Genotype to phenotype. Genotype to phenotype.
Genotype is what's stored in the cell, and phenotype is a physical trait. Right?
Yeah, so physical trait, I remember the pH, physical, pH to pH, phenotype to means physical, genotype talking about genes. Gene is a section of DNA that codes for one protein specifically. Yes. So we're moving on. 2 major parts are transcription and translation. We talked about transcription yesterday. These are the 3 things involved. We need messenger RNA, which is not up here yet. We need transfer RNA, which is right here. So how do you know, just by looking at this, that it is RNA?
Has a U.
Has a U, right? It has 1 strand, 2. Both correct. And we know it's tRNA. How to have you guys remember that? Looks like a T with some messed up, just kind of a messed up T. Alright? So, this, and obviously DNA is located where? In the nucleus, right? So those are the 3 types of things we need. We need messenger RNA, we need transfer RNA, we need ribosomes, and we need amino acids. So we just talked about this. This is review. Single-stranded RNA is. It has a ribose. You have uracil instead of thymine. So the 3 types of RNA. Messenger RNA, transfer RNA, right here, and ribosomal RNA. Ribosomal RNA is basically, it makes up part of the ribosome. Do you have to know that? No, you do not. Just make sure you know messenger RNA and transfer RNA. So the process in which, so we're talking about transcription now. Part of the nucleotide sequence is copied into a complementary sequence of our messenger RNA. So where does this occur? Transcription? Nucleus. Now, the reason why is this is real big. Because this is not a good picture of this, because, not a good model, because what am I missing with this structure, first off? What am I missing here with this DNA?
The backbone of the DNA.
The backbone of DNA, which is?
Sugar
Sugar-phosphate backbone. And also, what do we know about DNA?
It's in a coil.
It's a coil, can be if it's chromosomes, yes.
It's extremely long.
It's extremely long. So, in your first chromosome, you have 246 million of these. So this is made smaller just so you can see this on the board. Otherwise, this would be going from here and then all the way into the next room. So why does it occur? We have to get this information, because this is too big to leave the nucleus. This little line here represents the nucleus, and it's too big to leave there. So we have to make a copy of that. So we have this enzyme called RNA polymerase. So what's going to happen? RNA polymerase, and I'm going to represent RNA polymerase with my hands here, is going to come in and is going to unzip the DNA. What holds this DNA together?
Hydrogen bonds.
Weak hydrogen bonds, correct. So it's going to hold that together. So this DNA, this RNA, polymerase separates the DNA. So we obviously know that this is RNA because it is single-stranded and got U's. Doesn't have a T. So what's going to happen here is that RNA polymerase, remember my hand, is going to separate this. These guys are going to make copies, pop, 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 and RNA polymerase goes down in a sequence like this, and now we have a new copy here, and this what do we call this thing?
RNA.
Yes, specifically.
Transfer RNA.
Nope.
Messenger.
Right, messenger RNA. Because transfer RNA looks like a T. Messenger RNA. Now, the reason it's called messenger RNA is because this is much smaller than 246 million base pairs long on one DNA strand, right? So this can actually leave the nucleus. But what we just did right now, going from DNA to messenger RNA, is called what? Transcription. Now, the way I said it yesterday, yes, go ahead.
But why is it necessary to do this, to copy some DNA?
Because we're going to make a protein. This protein is going to go to the ribosome, magic's going to happen, amino acid chain, boom.
Gotcha.
We're going to get to that though. And the reason this can't leave the cell is way too large.
Yeah.
It just would be chaos in the cell. Now, how did I say yesterday to remember about transcription?
With the monks.
Like people transcribing stuff. When you transcribe something, you're copying something down. So I used the idea before the invention of the printing press. Monks or people had to physically handwrite and copy books out by hand. So they were copying it. So look what we did. We went from a base, went from a nucleotide to a nucleotide. So we're copying it. We're going from one nucleotide to another nucleotide. Alright? So that part inside this nucleus is transcription. We're copying it. Then messenger RNA is why it's called messenger. It's the messenger boy. Ribosome. So it leaves the DNA because it's small. It can go out of this. I will show you some animations, and it's going to blow your mind how fast this happens in your cells, in your life. So RNA is separating these. RNA polymerase, that enzyme. All right, and I'm making sure that we are still where we're at. So this is where we started off with our notes, correct?
No.
No,
You've already got translation?
yep.
Okay.
We're like halfway through how this translation.
Okay. Just let me know so I can remind you guys, like, oh, you've been taking notes now. This is all review. So inside the nucleus is transcription. Don't memorize the steps, understand the process, and it'll all fall into place. DNA can't leave the nucleus, it's too big. You got to make a copy of it. You got to have a messenger boy. And that messenger boy is messenger RNA. So this is messenger RNA right here. So what did I just cover up? What is the structure?
Ribosome.
Ribosome. Okay, so this happens on the ribosomes. So why does translation occur? Well, you want to make proteins eventually. You want to make proteins. So now we're where we need to be, right?
Almost.
3 or 4 down.
Okay, thank you.
Stop.
So messenger RNA leaves the nucleus and attaches to a ribosome. Once again, we said that. So that was what I just said here. This line represents the nucleus. So it's going out and it's going to attach to that ribosome right here. So tRNA carries a specific amino acid. That's why it's called transfer RNA. It carries a specific amino acid. So that's an amino acid. And what I'm having right here in my hand, these are amino acids. Does anybody know how many amino acids you have? How many there are out there, I should say?
16 or 19.
There's 20. There's 20 amino
Plus
acids.
that's
20. But you guys don't necessarily have to memorize that, but it's a good thing to know. There's only 20 amino acids. That's kind of astonishing when you think of all the different proteins there are out there. Okay, so that's why they're called transfer RNA, because they are basically carrying these amino acids. Because amino acids make up what? Makes up proteins, right? So we have the anticodon and the codon. We're going to get back to that here in a second.
This is where we left off.
Okay, the anticodon and the codon. So basically, the messenger RNA is going to be the codon. The transfer RNA is going to be the anticodon. Now, let's review. DNA. We all know this, correct? How do these pair up?
Beta A and GC.
A to T,
GC.
straight line, curved line, correct? That's DNA. RNA is what? Vowel,
Consonant.
and then somebody else had a different way of saying that, right? Looks kind of like almost like a lowercase a. Do you remember? So So those are this bonds with this bonds with this. So now, if I have messenger RNA and I'm in a ribosome, we eventually want to build a protein, correct? Proteins are made of amino acids. So what we need to do is so when I have this, A goes to what in RNA? U. This goes to, this goes to, oh, check this out. Transfer RNA is what happens in your cell, lines up right here, okay? And typically, what happens is this is going to be your code, and this is your anticodon. I will explain that here in a second. Alright, so that's the first one that happens. So, now peptide bonds form between the amino acids that are brought to the ribosomes. In this model here, that's the yellow things, the peptide bonds. So if I have GGC, what should that line up with? CCG, right?
So the glycine.
Yes. So here, this would line up like this. And then this nice little peptide bond would form. And now what actually happens is, this guy says, I'm done with you. That spits it out. And this kind of comes down like so. Okay, make sure I line this up. And you see how this is going to continue here, right?
So it just continues until there's just one transfer RNA and there's a bunch of amino acids kind of trailing it.
Yep, that's exactly what's going to happen. So the next one is U. I probably should cover it up. Like, which one is it? I'm like, yeah, how should you know? UCA, right? So it's right here. UCA. Boom, and no, that's not correct.
Yeah, it's the one right
Yeah, what's wrong with me? I said UCA, but I didn't grab the right one. So, UCA is right here. Boom. Peptide bond. And then this guy, once again, flop. Okay.
So this one, it gets rid of one?
Yep, it spits one, actually spits one out after it uses it. And actually, I think that the art, basically, to think about it very crudely, the ribosome's kind of like just, I mean, the ribosome just attaching it, attaching. And this is, if my hand's a long strand of messenger RNA, it's just going. Then outside of this ribosome is going to be this amino acid forming, amino acid chain, aka protein. So then we have, hopefully by process of elimination, CAG, right? That makes sense. Those are the base pairs. And then, boom, spits that out. Now, notice there's three more here. This is what they call a stop codon. We'll get into that later. This tells that, hey, we're done with the gene. We are finished with this gene. So it tells the ribosome basically to release this protein. And now we have a protein here. Because a protein is made of amino acids. Yes, sir?
So is this like a conveyor belt where the one strand of RNA just keeps on going until it, hits the end of it, and then the product, the amino acid, just
pops out, pretty much. And one of these days, I've decided if it's going to be Friday or tomorrow, I'm going to show you some really interesting videos. It'll blow your mind how fast this actually occurs. There's a real-time video I found that will really bring it home. So the process that we just did, we just formed a protein right here. That's what we just did. We just formed a protein. Now that process that we just completed out here, outside the nucleus, is called what? Translation. Translation. The reason it's called translation, how did I say that yesterday? Why did we call it translation? So the reason it's called translation is because we went from these bases, we're translating that to an amino acid. Henceforth, it's called translation. So it goes from this to this because we're translating that code. We're translating that code. Now we're going to talk about some definitions here. I'm going to explain this to you. I'm going to explain this process to you. A codon. So, in your notes, you'll need to write this down, is a 3-nucleotide sequence on the messenger RNA that codes for one amino acid. That's a whole mouthful, but it's really simple once you understand the process.
So a 3-nucleotide.
Oh, there it is.
So could you stop that for a second, Caleb? No, you know what? Don't. I'll just fill it with information because I don't want to have 2 videos up. So basically, you're having these codons and anticodons. So once you guys get done writing, I'll read it out loud. So a codon is a 3-nucleotide sequence on the messenger RNA that codes for 1 amino acid. An anticodon is a group of 3 nitrogen bases on tRNA molecule that are complementary to messenger RNA codon. This second is going to, that's confusing definition. It's perfectly, makes sense biologically. I'll explain it. So you have a codon, you have an anticodon. The important thing to remember is 3 nitrogen bases on each of them. They call this a triplet code. For every 3 nitrogen bases, code for 1 amino acid. And this is why. Because you have 3 nitrogen bases, 1, 2, 3, that code for 1 amino acid. That's why it's called a triplet code. So, everybody ready? We to discuss this? All you know is that ribosomes is what's where protein synthesis happens. We already know this, because I've said that at least probably 30 times since the school year started, I would bet. So please don't miss out on the UC.
What was the last part of the last slide? The last question.
Now I'm going to explain why this is called a codon, real quickly. Codon, anticodon. Remember, it is 3. So, 1, 2, 3. This is messenger RNA right here, correct? This whole thing is messenger RNA. So, 1 base, 2 base, 3 base nucleotides, okay? That codes for, that's called the codon. Now I'll explain why it's called the codon. It's not randomly named codon to be mean. It's coding for an amino acid, right? That's the code for it. Now, to decode that, to get the right, to turn that into what we want, we need the anticodon. So the anticodon is going to be this. 1, 2, 3. 1, 2, 3. So for every 3 bases, it's going to code for 1 amino acid. That is what that means. So all you really need to remember is that tRNA is talking about, we're talking about anticodons, and messenger RNA, we're talking about codons. You need to know that it's 3. It's a 3-to-1 ratio. Because of this nice little transfer RNA, it can only take 1 at a time. That's why it's a codon. That's why it's 3 for every 1 amino acid. Questions on that? Yes?
Is there any particular reason why it's shaped in that weird way, or is that just how you cut it out, or what?
No, and actually they looked at the molecular structure of it. That is analogous to how it's shaped, if the molecular structure of it.
Right, but is there any real reason why it's got those 2 weird bumps?
Yes, it probably does, and the actual rationale behind that, I'm not sure. But it is specifically a reason I would imagine has served some function. So basically, I don't know what it's made of besides the transfer RNA, besides these 3. There are other parts of this. I just can't remember. It's been a while since I've gone that deep into it. But they probably does serve a function. But what's nice for us is that this kind of helps us make it look like a T, so you can help remember it. So, questions on that? No? All right. Let's see, was that it? Hold on, wait. Okay, go ahead and turn that off for me. Thank you very much. I appreciate it.